seasonality is a really critical component of ecological adaptation. Most organisms on Earth, at least those in those biomes, experience fluctuating conditions across the year that they have to cope with. And so we expect that natural selection should act strongly to synchronize the life history of species to be synchronized with their abiotic environments and with their ecological communities. Seasonal adaptation is also really important perhaps for speciation, and that's because it can provide a direct link between ecological adaptation and reproductive isolation. So if ecological adaptation requires a shift in phenology, because reproduction itself is often strongly seasonal, that could result in a direct effect on systems theory. So in this way, life history adaptation and seasonality can serve as a magic trait for ecological speciation, providing a polytropic between ecology and mating systems. And indeed, phenology has been invoked as a really important reproductive isolation trait in a number of ecological incipient speciation stories. Along with speciation, it's also really important for adaptation to climate change. So changes in phenology are expected to be and are already really observed to be some of the major impacts on ecological systems from global change. And so the ability of insects, or organisms more generally, insects and kids this dog, to deal with these shifts in phenology evolutionarily is really important to understanding how communities are cope with global change. So today I'm going to talk about the role of life history adaptation and seasonality in one of the kind of textbook cases of ecological speciation, Rankine's Powell. Yeah, so this fly is a differential fruit fly that specialized in infesting the fruit of downy hawthorn trees in eastern North America. The flies lay their egg in the fruit, which hatches into a maggot, feeds within the fruit until it drops from the tree. Then the egg crawls out, they pupate in the soil. These insects only have a single generation per year, so the vast majority of their life cycle is spent in this pupal stage, in dormancy, a dormant stage called diapause in the soil, about 80% of their lives. And they stay that way until the following year, when they close out as adults, right before hawthorn fruit gets ripe again in the early fall. And all this was going along fine for tens of thousands of years until settlers from Europe introduced apples into North America in the 1600s. And then sometime in the mid-19th century, a population of these flies shifted over from infesting hawthorn trees to this derived apple host. Now, this ecological transition to apple required divergence in two key ecological traits. The first is their differences in olfactory behavioral response to fruit follicles, which is how these flies find their host plants, and ultimately the flies with which they'll mate. And the second trait that's going to be the focus of the talk today is their life history timing. So apple trees and hawthorn trees have different fruiting phenologies. When they overlap together, hawthorn flies tend to set fruit about a month later than apple trees. And these flies, because they only have a single generation per year and they have a relatively short adult lifespan, they have to be tightly synchronized with this ephemeral resource. And indeed, the apple flies come out in the field about a month before the hawthorn flies do. Because they don't live long, this really limits their ability to breed with each other. So both of these traits together result in about a reduction of about 4% gross migration between sympatric populations of apple and hawthorn flies. And this 4% is actually just kind of the surface of reproductive isolation here, because strong ongoing selection against F1s and hybrids actually drives strong post-zygotic isolation as well, which contributes to much greater amounts of reproductive isolation than just this 4%. And this is reflected strongly in the patterns of genetic differentiation of these flies. And we know that there's widespread associations with life history timing across all 5 chromosomes of these flies. This is data that's derived from a 2010 microsatellite study that showed that a number of the loci that are really involved in differentiating these flies in a population genetic sense are also strongly associated with life history timing in these flies. And now that we've gone on to look at this with rad-seq data and thousands of SNPs, these patterns are even more apparent there as well. So we have a case where we have this widespread association with life history timing that suggests that this trait that's involved in reproductive isolation is highly polygenic and really quantitative. And that's the angle from which I've become interested in understanding life history timing in these flies, is trying to understand the relationship between the complex patterns of genetic variation we see in the genome and this big picture ecological trait driving reproductive isolation. So here's sort of a summary of the life history of these flies. So in red here is the little narrow window of ripe hawthorn season, and in blue we have winter, which for the purposes of these flies is really the time at which temperatures are below their physiological thresholds. And so before I said that these flies only have one generation per year, and most of it's spent in pupal dormancy and diapause. So when flies were able to use this new resource of apples, when they exploited this new niche, they couldn't do it by just tweaking one portion of this life cycle. Rather, they had to reset their entire life history in order to be matched up with this new resource. And this is important in a number of ways. This is sort of a linearization of those cycles before, with apple flies up here, hawthorn flies here. This highlights how there's really kind of two critical time points that were needed for adaptation to this new niche. Both differences in pre-winter life history and post-winter life history. So in pre-winter, these two fly populations are entering dormancy and pupating under very different conditions. For the apple flies, it's really in the dog days of summer, whereas hawthorn flies a month later, right at the onset of frost in the upper Midwest. And most of the work on selection and life history timing in Rhagoletes flies is really focused on this pre-winter part of the story. But it's the post-winter part of the story that really drives the difference in phenology that's ultimately driving prezygotic isolation here, that these flies come out at different times post-winter. And that's what I'm interested in tackling here. So the broad questions that we're trying to address here are what's the functional basis of this difference in post-winter life history timing between these two flies that drives your critical isolation. And we're looking at it from a functional perspective because physiology is really the interface between the genome and the environment. And we're trying to draw a better map between the complex genomic variation we see in this big phenotype by breaking the ecological phenotype down into its component intermediate physiological phenotypes that we can then use to make better connections to allelic variation in these flies. One of the first things we wanted to do was to look at exactly what's different between these two flies. When do they start to diverge developmentally post-winter? And because they're in opaque puparia, we can't watch what they're doing, but we can infer what they're doing by measuring their metabolic rates. And that's what these data show, that we have individual metabolic trajectories for apple flies on the top and hawthorn flies at the bottom. And what we can see here is that apple flies really terminate diapause, which is indicated by this release from metabolic suppression really early after you pull them out of the winter. The next thing I wanted to do with this was compare the different parameters that make up these nonlinear models to figure out what's exactly different between the curves for both of these populations, and then understand what part of those curves is actually related to implosion timing as well. And we got a really clear answer for this, for both of those questions at all, and it's the parameter that drives this initial increase. This diapause termination is really the only part of these curves that are different between the two flies, and it explains almost all the variation in implosion timing. So it's this lift from metabolic suppression, termination of diapause, that then leads to the difference in timing between the two flies. And the simplicity here might seem a little bit at odds with that idea of this being a big quantitative polygenic trait. We only have a single component of post-winter life history that's involved here. But diapause termination itself is not a monolithic trait. Rather, it's serving as a hub with a lot of inputs from different upstream physiological processes that then flow through this hub to eventually affect variation in life history timing for these flies. Let's back up for a little bit to talk about exactly what diapause is. So diapause is not a static state of suspended animation. Rather, it's a dynamic alternative developmental pathway where normal development, morphogenesis, and metabolism are suppressed. And diapause is really widespread across insects, arthropods, and a lot of groups, but it's not very well conserved. Closely related insects differ strongly in their capacity for diapause, they differ in the life stage in which they undergo diapause, and they differ a lot in the token environmental cues that drive the initiation and termination of dormancy. But despite all those differences, there are some important commonalities, and the most important one is that insect diapause is almost always driven by a suppression of ecdysteroid signaling. Ecdysteroids are the hormones involved in molting in insects, and they're the ones that control the progression of development and metamorphosis in these flies. And this really highlights how diapause termination can serve as an important hub here. So we know a lot about ecdysteroid signaling, and we know that it's a neuroendocrine process. And it's this that's really driving the ultimate life history time of these flies through activity in the brain and associated neuroendocrine structures. And so if we look at just the upstream steps for diapause signaling, there's a lot of different places where variation, allelic variation, could be acting on the end result of your diapause signaling. And if we go upstream from that, from the things that are going on in the brain and these neurosecretory structures before PTTH synthesis, there's really a myriad of different input points where allelic variation could be affecting the overall outcome of diapause termination. So to understand this, the first RNA-seq study that I'm going to talk about today is one that's led by Pete Myers that actually came out in Journal of Experimental Biology yesterday, where we looked at the pupil head transcriptomes of these flies at 3 different time points after 5 months of being in the fridge. One when they were still cold, when they were still in overwinter, and then 2 time points after they warmed up. And the purpose of this was to understand how the apple flies are getting to be more potentiated to terminate diapause quickly right after winter. One hypothesis could be that they're just responding to environmental cues in different ways, that the differences that we see in them are elicited by this shift from cold to warm, or they could be accumulating differences during diapause in their brain. And what we found here is, first, not surprising, when you take a pupa that's been cold for 5 months and warm it up right away, you see massively upregulated gene expression across the board. But along with that, there was really a strong amount of differentiation that predated the shift to warmth for these 2 flies. There's about 3,000 genes that are differentially regulated between apple and hawthorn flies while they're still in overwinter at this time. And when we look at what's actually differentiated here, we see that a lot of what's going on is that genes involved in development, morphogenesis, and cell proliferation are kind of going up all over the place in apple flies early. And it's not just in the brain, it's in a lot of other places. It's the imaginal discs, tracheal development, what's going to become the adult cuticle, and all these things are upregulated outside the brain. So what we really found here is actually downstream of what we're really looking for. That the apple flies, even though we don't see a lot of morphogenesis at this point, they're really geared up to, all their tissues are really going to start developing right away. So in order to kind of hone in on this more, the next thing that we did was try to get rid of all the distracting things going on in the head and focus on just what's going on in the neuroendocrine transcriptomes themselves, with the brains, subesophageal ganglia, and this little very delicate but important structure, the ringware. And what we did was use these brains and step them back through a careful time series. We started at a time point where these flies are already really strongly differentiated from each other, and then walk backwards through time and figure out what are the very first gene networks involved in differential diapause regulation in these flies. And we did 5 time points. We had to pull 20 brains to get enough raw DNA, so there were 1,000 random brains that went into the study. The results for this are actually really kind of still hot off the servers. I've just had the raw reads in hand since May, but so far we're seeing some really promising things that we've actually done when we set out to do here. For one, it is much more tightly focused resolution. Instead of 3,000 genes differentially regulated, we have a few hundred at each of these time points. And what seems to be going on here, based on the initial results, are that there's active brain development during diapause in these flies, and apple flies are accomplishing this much more frequently. So to look at some of the example genes that we see upregulated in apple flies, is things that we were expecting to see. Things involved in central nervous system development, a lot of Wnt signaling genes, axon guidance, and global transcription all seem to be going up in the apple flies very, very early in diapause. But it's not just a case of things going up in apple flies and staying static and transcriptionally silent in hawthorn flies. We actually see a lot of active suppression of these same things in hawthorn flies as well. And with that, in time, I'm pretty much done, so that's it.